In this module, we will study the affinity chromatography, another important technique for separating the biomolecule. As the name implies, that theoretically affinity chromatography is capable of giving absolute purification even from complex mixture in a single process. Since this technique depends on the affinity or on the property of attachment or what the affinity a biomolecule has for a support, so it means that it can be absolute. It can give a very high purity of the compound present in a complex mixture in a single process. Now, this technique was originally developed for the purification of enzymes, nucleotide, nucleic acids, and the immunoglobulins, which are important biomolecules for a molecular biologist. Now, membrane receptors and even to whole cells and cell fragments can also be separated by this technique. And what is the basic principle is that the technique requires that the material to be isolated or separated is capable of binding reversibly to a specific ligand. The important thing is that that material should be capable of binding reversibly. Reversibly means that not permanently but temporarily, so that it can be reversed to a specific ligand, specific compound that is attached to an insoluble matrix. Insoluble matrix means a support and the material is attached with that support and then that substance is attached to there. For example, there is a macromolecule, then the ligand, and it can reversibly attach and detach with that molecule presently. The technique can be applied in two distinct ways. There are two distinct ways with this technique can be applied. One is compound will bind to the ligand. That is the compound which we want to separate should bind to the ligand, to the chemical. And all other compounds, they can be therefore be washed away. Because the compound which we want, that should be bound to the ligand, whereas the other compounds which are present, they are just washed away and the compound subsequently recovered by displacement from the ligand. That is, it is attached, then it is detached from the ligand. As shown in this diagram, you can see the matrix, then there is immobilized ligand and the enzyme. It attaches with that, goes over there with a solid support. It holds the enzyme. Then there can be two ways. One is either it can be dialysis, it can be dialyzed, and we get the pure enzyme. Or the second is that we can wash it with a certain compound or the non-specific elution which splits it, and the pure enzyme that is recovered at the end. So this is, in the matter of fact, the principle of purification of enzyme by affinity chromatography. Similarly, we can use a chromatography by for other compounds as well. Now, an ideal matrix for affinity chromatography should have following characteristics. Like, that should possess the suitable and sufficient chemical groups. Because the chemical groups, the ligand, they are the key factor in this separation. Then they should be stable during binding. That is when a compound, as a matter of fact, is there and bound, that should be stable. It should remain over there, and the micromolecule that should be attached to it, so that subsequently those micromolecules like proteins, nucleic acid, they can be further eluted. Interaction, only it should be capable of interacting only weakly with other macromolecules to minimize the non-specific adsorption. That is, if there are enzymes present, there are nucleotides present, and other compounds are present, then it should be able to interact only weakly with other compound rather than the major exhibit or the major compound which we are using. Then it should also exhibit a good flow properties. Because we have to elute it, then it should be very having a property, a good flow of the material through that column. Now the ligand, the chemical nature of a ligand, that is directed by the biological specificity of the compound to be purified. That is the chemical ligand or chemical nature of the ligand which we should use for that compound. That should be for the biological specificity required for the biomolecule. It should be absolute specific for that biomolecule, and it will bind exclusively to one particular compound. That is the protein or the nucleotide which we want to separate. The ligand should be capable of binding with that. Then it should also be capable of group selectivity. That is, it should be able to select the group, and it will bind to a closely related group of compound that possess a similarly inbuilt chemical specificity. That is, it should be group selectivity, it should be capable of having a group specificity over there. Now in this table, I have tried to demonstrate or put that what type of different group or ligand we can use for separating the different compound which we often come across. Now its application is, as shown, many enzymes and other proteins, including receptor proteins and the immunoglobulins. They have been purified by affinity chromatography. That is, this technique is very useful for further purifying the material. The application of the technique is limited only by the availability of the immobilized ligand. You remember that this technique has got a drawback, that its application is limited only by the availability of the ligand which are available to bind those compounds which are present, and the principle have been extended to nucleic acid and have made a considerable contribution to development of molecular biology. So as we have seen, that affinity chromatography has played a major role in the molecular biology science for separating the molecules, biomolecules, membranes, and other enzymes which a molecular biologist frequently come across and uses for further investigation.